And we'll wait for the ding dong. I wanted to talk a little. This is not a hugely long presentation, but we talked about fisheries on Friday. Guess that's appropriate that we had fish on Friday. And I wanted to talk about other things that we get from the ocean besides just raw seafood. And one of the things that we get is biological products. That includes everything from cod liver oil, which I don't think is so popular now, but it used to be widely used as a vitamin D supplement, to the various, omega-3 shark oil and fish oil supplements and things that you can take. But there's also pharmaceuticals that have come from the ocean, even though they're usually not directly harvested from the ocean itself. It is, what are you doing here? Not you guys. Sorry, I've got a cat that has decided that, sorry, my cat has decided that he's my best friend and my shoulder is where he wants to perch, and he's dug in his claws in a rather uncomfortable fashion. Anyway, if I can get through the lecture with this cat sitting on my shoulder, let's look at chemical compounds that we've been able to get from ocean creatures. If you're a sponge, you can't move. Well, some sponges can crawl, but the fastest recorded speed for a sponge is about 4 millimeters per day. So you're essentially stuck where you are. You are made of digestible tissue. If you're a sponge, you may or may not have a skeleton of sharp little pieces of calcium carbonate or silicon, but certainly fish with hard mouths can crunch you up right there. And so you need something more than just sitting there and looking pretty if you're going to survive on your typical coral reef. And it turns out that sponges are an incredibly rich source of bioactive compounds. You've probably heard that we get a lot of medicines from plants. Something like a quarter of all prescription drugs are either isolated from plants or at least were first discovered in plants, although we might make them by using some other technique. Well, it turns out the marine biosphere is no different. Sponges and other organisms, especially the ones that need chemical defenses, crank out an enormous diversity of chemical compounds, and it's turning out that many of them have antibiotic properties, or they're antifungal, or in some cases anti-cancer. And this is one of the frontiers in drug discovery right now. People are looking at the ocean realm. And I've just heard this thing go ding dong, so let me let somebody in. Here, okay, admit. I will temporarily, here, you get off. Since we paused, this is the cat that was up on my shoulder that sank his claws into me. This is Jackwagon. Yes, you're a nice kitty, sort of, for some definitions of the word nice. Now, go somewhere else. And that was your dose of gratuitous cuteness for the day. So getting back to the presentation. Yeah, cat's adorable. He's adorable. He'd be a lot more adorable if he didn't feel the need to sink his claws into you, not out of meanness, just out of, I can't understand what I'm doing wrong, why these people keep bleeding on me. He's just kind of an idiot. Anyway, most of these compounds are only produced in minute quantities, though. The problem is that typical sponges might only have, picogram amounts of such and such an interesting compound. So if you discover something bioactive in a sponge, or for that matter, a soft coral or anything else, the challenge is often to come up with a way to synthesize it, to make it under controlled conditions from simpler feedstocks that don't require you to grind up tens of thousands of pounds of sponge in order to get, just a few doses. In some cases, this isn't possible yet. This is a sponge called Disidia avara. There's a rope across your field of view running from left to right. And in the center of the picture, you can see three lumpy things that look a little bit like golf balls with a hole in them. Those are the sponges, and they're growing on a rope. This is a fairly common way of culturing sessile invertebrates. Oysters, for example, can be cultured on ropes. And these sponges are being cultured in the Mediterranean Sea because they produce a promising drug called Averol that I think is not yet approved and is still in testing. If it is approved, I assume someone will try to figure out how to make the stuff in the laboratory, but it may be necessary to culture an awful lot of sponges for a while. I'm not sure what the current status of that is. Sometimes, though, we get ideas, even if we don't get an actual drug. A scientist named Werner Bergmann started doing chemical analyses of some Caribbean sponges back in the 1950s, including this lumpy thing called Tectitethia crypta. And he found that Tectitethia contained things like this. This ought to be recognizable. It's recognizable as a nucleoside, right? You've got a base over on the upper right, that double ring of carbons and nitrogens. That's adenine, I believe. No, it's not adenine. I think it's okay, it's something. It's a base. And below to the lower left, you've got a sugar. You've got good old ribose. And then that hydroxyl group over there on the far left could easily be stuck to a phosphate group, and you'd have your basic nucleotide. However, notice that one of the hydroxyl groups on the sugar is pointing in the wrong direction. If you actually tried to feed this to a cell that was trying to use it for probably transcription, it would jam the machinery like a misshapen staple jams a stapler, right? If you're giving your cell something that looks a lot like a nucleotide but doesn't quite fit, you can confuse the heck out of the little replication enzymes and shut down DNA replication or possibly RNA transcription. Now, these modified nucleotides that Bergmann found were called spongothymidine and spongouracil. Those specifically turned out not to be particularly useful as drugs. But the basic idea of shutting down DNA duplication or RNA transcription by dosing a cell with something that looks almost exactly like a nucleotide but isn't, that idea has led to some of the most fruitful drug discoveries that we have. A modified version of one of these sponge nucleotides is called vidarabine, and it's been used as an anti-cancer drug, and it's also an antiviral. Think about it. Cancer is out of control cell proliferation, and you can stop cells from proliferating if you can shut down their ability to synthesize DNA. And then viruses you can shut down if you can stop a cell's ability to make DNA or possibly RNA. So vidarabine has been used both against cancer and against viruses, notably herpes. And vidarabine was the first of these nucleotide analog drugs, drugs that block nucleic acid synthesis by resembling a nucleotide but with modifications to them that stop DNA polymerization. And there's a lot of other examples. One of the ones you see in the picture here, down towards the lower left, is AZT, or zidovudine. That is a thymidine nucleoside, except it's got a nitrogen in the damnedest place. That blocks DNA chain elongation, and that's an anti-HIV drug. It was one of the first to be developed, in fact. They developed that I know they were giving it by the early 90s. Ditto for some other anti-HIV drugs like DDI, dideoxyinositol, and DDC. Other antivirals are in this class. Acyclovir is a big one. That's an anti-herpes drug. Remdesivir is yet another antiviral. That one's been in the news late because it got approval for treatment of severe COVID infection. Donald Trump got it, for example. I think the word is that it doesn't do much good for most infections, but it does seem to help in really severe ones. I'm not up on the very latest developments there, but that is an antiviral drug that is one of these nucleoside analogs. Ditto for some anti-cancer drugs like gemcitabine and cytarabine. So that principle of shutting down DNA replication by dosing it with slightly misshapen nucleotides has led to an awful lot of drug discoveries. There's lots of people alive today because somebody got the idea to do chemical analyses of a sponge. So there, maybe you'll look at sponges with more respect. Yeah, you know, I used to have a theory about sponges, but unfortunately it was just full of holes. Anyway, this is a marine bacterium, and it belongs to a group called actinomycetes. They are very common in soil. In fact, actinomycete byproducts called geosmins are responsible for the smell of soil. You know, I don't know how many of you are into gardening, but if any of you have ever dug up a nice, rich shovel full of earth and get a good sniff, what you're smelling is largely due to the actinomycetes in the soil. And soil actinomycetes have produced antibiotics, streptomycin in particular, nocarticin is another, and their analogs. But there are some actinomycetes that live in the ocean as well, and this one's called Selenospora. I should mention actinomycetes have this superficially fungus-like growth form. They form long filaments of cells that'll branch off and give rise to spores. So actinomycetes are convergently similar to fungi. You know, black bread mold, for example, is vaguely similar to this. And Selenospora was only discovered in 1989. There's not a huge number of people who are going out and looking for actinomycete bacteria in the oceans. Selenospora pacifica was only named back about 14 years ago in 2006. Why should anybody care? It turns out that Selenospora makes a freaking medicine chest worth of bioactive compounds, and this is one of them. It's called selenosporamide A. I don't know the current status of it, but last I looked it up, it was in clinical trials right now as a drug against one of the worst forms of cancer you can get, multiple myeloma. This is one that attacks the bones. And I don't think we have really good treatments for it. There are some cancers that we've got better treatments for and others that are much less tractable. But this one is, at least as of now, showing promise. So there's another compound. that looks promising as a medication. What you're looking at here is called a bryozoan. Bryozoans are, oh, how should I describe them? Individual bryozoan animals are about the size of a pinhead. They've got a gut, they've got little feeding tentacles, and each individual lives in a roughly squarish little box called a zooecium. The thing about bryozoans is that virtually all of them are colonial. Each individual bryozoan might be the size of the head of a pin, but each individual can give rise to new individuals by budding. Each individual can send off a little stalk, which can sprout up a new individual, and that one can send off a new one, and so on. Almost like kind of like crabgrass propagating by runners. And colonies of bryozoa can get pretty large. And this is a colonial bryozoan called Bugula. Sounds like a good name for a vampiric insect. And in the 1960s, a compound isolated from Bugula called bryostatin turned out to be an anti-cancer agent. At least in the lab, it killed cancer cells. If you're wondering what the size is right here, the Bugulas that I've handled, each one of those spiral-looking things that you're seeing would probably be about 6 inches across. So your field of view here is going to be roughly 2 feet. Bugula is really common in the place I've seen it has been in the Gulf of Mexico, growing on pilings and docks. There's lots of Bugula. It turns out that bryostatin wasn't useful enough relative to risk. It does have some anti-cancer effect when you give it to people, but the benefit that you get is not large enough relative to the side effects and the risk that you suffer. So it's not been approved as a standalone anti-cancer drug. There was interest for a while in using it as part of a chemical cocktail. These days, a lot of cancer patients are treated not with a single anti-cancer agent, but several all at once. And the idea is that you try to hit those cancer cells from several directions at once, interfere with several different cellular functions at the same time, and really get them in the crossfire. I've heard of bryostatin being used as part of this chemical cocktail. How well that works, I don't know, but I do know that bryostatin is being tested now as a treatment for loss of synapses, which is one of the processes that makes Alzheimer's disease so bad. So far, it looks promising. I can't promise that this is going to be, a cure or even an effective treatment for Alzheimer's disease, but it's certainly worth checking out, and people are checking this out right now. One more cool example. On the right, you've got the shell of a marine snail from the South Pacific called Conus textile, Conus textile, I guess, because the pattern on it reminded somebody of some kind of fancy fabric, some kind of fancy textile. It's called the textile cone. And the animal is a predator, and you would not think snails would be very effective as predators. It's not like they're going to chase anything down. But these, in fact, have a proboscis that contains these harpoon-like teeth that the snail can eject, that the snail can literally shoot out and harpoon its prey. And on the left, that's a scanning electron micrograph of what one of these radular teeth looks like. This wouldn't be exciting, except these snails also secrete venom, a rather complex cocktail of neurotoxins. Snails that specialize on feeding on other invertebrates like worms are not particularly dangerous to humans. I mean, they can sting you like a wasp can, but it's not particularly bad. But some of these cone snails are adapted to take down fish. And the problem is that if you are a snail, you're not fast, your body is very soft, although the shell might give you some protection. The fish is probably stronger than you are and definitely faster than you are, which means that what's adaptive is to put that fish down as fast as possible. You know, the same reason why if you're, hunting tigers or something like that, you might only have one shot, and so you carry this seriously overpowered rifle. Um, Mind you, you should not be hunting tigers unless they're a direct threat to humans because they belong living free and in the wild, like the Krat brothers say. But if you ever have to put down a dangerous tiger, big game hunters carry these ginormous rifles or shotguns with serious stopping power. It might be more sporting to try to hunt a tiger with a 22, but you might get killed. And it's a similar case here in that that snail has to put down its prey as fast as possible. I almost said as fast as humanly possible, so I guess as fast as snailily possible. And the venom is extremely potent, and it's dangerous to humans. There are people who have died from getting stung by venomous cone snails. Naturally, this happens in Australia, although to be fair, there are other parts of the Pacific where these cone snails live. And that's because, and they're dangerous to humans because fish are a lot more close to us than worms are. So a venom that puts down fish in seconds is going to have a very similar effect on humans because we're chemically more like fish than we are like worms. Why am I bringing all this up? This species is called Conus magus, and its venom has been studied. It's turns out to be a whole cocktail of different toxins called conotoxins. They found one in particular that turns out to be a short peptide. I think it's 7 amino acids in a row. And it turns out that this can be used as a non-addictive, a non-habituating painkiller. Non-habituating means you don't build up a tolerance to it and require doses to get the same effect. Your body doesn't get used to it. It's called ziconotide. The trade name is Prialt. And unfortunately, you can't just take a pill with ziconotide in it. It won't have much of an effect. Even injections don't work very well. The way you administer this stuff is you have to implant a pump under the skin of a patient and have a little tube that delivers Prialt directly to the painful area. Typically damaged nerves. This is clearly not the kind of thing you would want to do for just a migraine, but it's very useful for patients with chronic pain, people with severe injuries to one of their limbs, where they may be in constant pain because of nerve damage. Prialt is very useful there. So it's not the kind of thing you're going to get at the drugstore anytime soon because you have to get one of these pumps surgically implanted. But there is a population of patients for which this is very useful indeed. And there are something like 300 species of cone snail out there. Certainly that's the logical place to look if you wanted to find more similar drugs that might have medicinal use. And maybe there's one out there that you can take as a pill or a simple injection. So we shall see. One more benefit that goes beyond just looking at drugs is a lot of marine organisms are very useful for very basic research. There's a reason why major discoveries in basic biology tend to get made at marine laboratories. A lot of marine organisms have features that make them useful for studying very basic stuff. Um, Simple example, a lot of research in the development of embryos has traditionally been carried out at marine labs because invertebrates like sea urchins and sand dollars and certain segmented worms, it's easy to get lots of eggs and sperm out of them, and they're fairly easy to raise in the laboratory and study. So, for example, one of the first really noted African American biologists, a man by the name of Ernest Everett Just, discovered that there's two different mechanisms that an egg uses to keep itself from getting fertilized twice. There are mechanisms called the fast block and the slow block. And he discovered these looking at sea urchins, but the same basic mechanisms apply to you. I don't know how universal they are, but they're pretty widespread. And he was able to discover them studying sea urchins. A lot of very basic developmental biology work focusing on questions like how do the cells know to do this, to divide or to move, to build these structures. A lot of that has been done on sea urchins and sea squirts and critters like that. And every so often we get some interesting insight into human well-being. You might have seen this slide before. These are the stinging cells, or not cells, they're the stinging structures of cnidarians, jellyfish and sea anemones and soft corals and things like that. Each one of these contains a little barb and a filament attached to the barb. On the left, you can see one that has not fired, and you can see that coiled up filament. On the right, you have two, one of which has not fired, and you can see the filament. And then below, there's one that has fired, and it shot out its barb. There's a diagram of a nematocyst in the process of firing. There's a little operculum, a little trapdoor that flops open when the nematocyst is stimulated. And the penetrator shoots out and then rapidly turns itself inside out, penetrating the skin or the exoskeleton of the prey and shooting out a little tube that delivers the toxin. The whole process may take less than a microsecond. There's some YouTube footage of this if you want to see what it looks like. So there's a nematocyst that is fired. Typically on each cnidocyte, that's the cell that contains the nematocyst, there will be a bristle-like structure called a cnidocyl, and the cnidocyl has mechanoreceptors. The mechanoreceptors sense vibration, and the nematocyst will fire when it's vibrated at a particular frequency. What's so great about this? The point is that these stings are kind of expensive for the jellyfish to make, and it's not beneficial to the jellyfish if it fires those stings when it touches a rock, right? It's only really beneficial if it fires those stings into something that it can eat. So very often we find that these cnidocyls are tuned to fire at a frequency of, I think typical would be 25 to 30 per second, 25 to 30 hertz, which happens to be about the speed at which little swimming shrimp tend to beat their little swimming legs. The cnidocysts also have chemoreceptors that will trigger firing in the presence of chemical signals. Something inert like a rock or a piece of glass or something like that won't make these things fire. Something that's made of nice, delicious proteins and things like that will trigger the firing. If we look at critters like sea anemones, they're covered with things that look like this. What you're seeing here is every cell has a collar of little finger-like extensions called microvilli that surround a single cilium in the center. So there's one cilium in the middle of that, and then surrounding it, there's a whole cone of little finger-like projections, little microvilli. And you can see that there are a bunch of these, and they're dead ringers for these structures, which are not on sea anemones at all. These are the hair cells that you use to perceive sound. They're located in your inner ear, inside a coiled cavity inside the densest part of your skull called your cochlea. The cochlea is buried deep in the thickest part of your temporal bone. Your temporal bone. That's the bone around on the sides of your head. The really thick part just behind your ears is where it's located. It's called the petrosal part of your temporal bone. And vibrations from the outside are transmitted from your eardrum through the chain of conducting bones, your hammer, anvil, and stirrup, malleus, incus, and stapes bones. And they set vibrations going in the fluid inside the cochlea, and that vibration is transmitted to these hair cells. They have the same basic function as in the sea anemones. They detect vibration at particular frequencies. This is probably something that we share evolutionarily with the last common ancestor of us and sea anemones, which would have lived something on the order of 600 million years ago. And the cool thing is that Dr. Glenn Meyer and his students and colleagues at the University of Louisiana Lafayette, my hometown, yay, go Cajuns, have been using this as a model system to provide insights into deafness. It's very hard to study human hair cells, because they tend to die if you try to take them out of the body. They can't be cultured for long periods. And B, if you take them out of somebody's ears, they go deaf, and there's not a lot of people that are willing to let you do that. And they don't stay alive very long after the death of the body, so you can't get them out of cadavers or anything like that. Sea anemones, on the other hand, have cells like this all over their tentacles. Sea anemones grow easily in the lab. They'll regenerate tentacles if you cut them off. These hair cells are right out there on the tentacle surface. They're not deeply buried in the thickest part of your skull. And so Dr. Meyer has been using these as a model system to look at certain types of human deafness and look at, what sea anemones use these for, and then look at things that might stimulate growth of cells like this in anemones and see if that transfers over to the human inner ear. I can't say that he's got a cure for all types of deafness yet, but it's fascinating work, and somewhere down the line, it may well lead to a breakthrough that will make somebody's life far easier. Hair cells tend to die if they're exposed to sound that's too loud. This is why you can go increasingly deaf if you're exposed to a lot of loud sounds. Construction workers, people that use jackhammers, rock guitar players tend to have some degree of permanent hearing loss. You can't regenerate your hair cells, but if anemone research could help us figure out how you could, then we'd have a cure for certain types of hearing loss, and that's worth pursuing. And that's something that we get out of the oceans. So there's a lot more that we can fish up for human benefit than just the fish that we eat. All right, and by the power vested in me, I am going to stop recording.